Welcome to GFC Diagnostics. We're a small, innovative company based near Oxford that has a novel DNA probe-based technology for rapid detection of bacteria. I'm Bruce Savage. I'm the project leader, and I graduated in life sciences and worked for about 6 years at Roche, the pharmaceutical company. I then spent a lifetime in the medical diagnostics industry before forming a molecular DNA probe-based diagnostic company called C.difficile back in 1991. Then in 2007, Graham Cope and myself co-founded GFC Diagnostics, and Dr. Cope will go on.
Hello, I'm Dr. Graham Cope. I'm the Technical Director of GFC Diagnostics. Some years ago, while I was at the University of Birmingham, working on smoking and smoking-related diseases, I wanted a test to improve my data collection of how people were smoking. After some investigations, I came across and developed the SafeTube. SafeTube comes in two parts: a volume collecting vessel for smokescreen, it's either urine or saliva. The second part is two chambers, one in the bottom for a mixed range of chemicals and two in the middle which contain highly reactive chemicals that have to be isolated. The test works by taking up the sample for urine 2 mL, the cap goes on, it's snapped on, which breaks the compartments, and the sample pushes through all the reagents and then they're dissolved in the fluid as it's reacting. In the safe smokescreen, the test turns pink if cotinine is present, which is a breakdown product of nicotine. Much later we were developing other tests for looking at other antibiotics, and we're developing a test for dapsone, a treatment for leprosy. I've been working with various aspects of GFC Diagnostics, and we came across Dr. Graham Mock, who's an expert in DNA bacteriology, and it's with his work that we're developing the test further.
I'm Dr. Graham Mock, did my PhD in organic chemistry at the University of Cambridge and spent several years in America as a postdoctoral fellow. Since then I have worked for almost 30 years in the biotech industry developing diagnostic tests, and very recently developing the ELOSA test. In this ELOSA test I developed a cross-linking method which led to the actual process being much more efficient and high yielding and capable of detecting very low levels of DNA.
Hello, my name is Alex Savage. I recently have graduated from Newcastle University, where I studied chemistry and environmental sciences. With my studies I learned a lot of analytical and scientific skills that I've carried forward into my job here. My role as part of GFC Diagnostics has been to help Graham Mock reformat his technology and apply that to our GFC's SafeTube system. That's meant a lot of experimental data collection as well as development of the assay. In particular, we've looked at operating freeze dryers and using that in the development process to improve upon reproducibility of the test and stability of the assay as well.
Hello, so this is GFC Diagnostics MRSAScreen trial test kit. This is to show what we'd be working towards as a sort of final product. It would be a kit like this, for instance, where we've got everything required to carry out the test in this box here. Alongside, this is a mock-up of what we'd want to achieve, which is a handheld portable device that's battery powered. So let's have a look to see what's inside our demo kit here.
This is the kit. Open it up. It comprises up of 3 parts. We've got target, wash and colour. This is done chronologically, 1 through to 3. Then unpack one of our tests. See, we have a freeze-dried probe, strep-tabbed coating at the bottom. You start the test with first the target phase. Comprises up of sample bottle and a syringe.
We draw up this solution inside the sample with the syringe provided. A set volume is taken, and that set volume is then plunged down through into our test. And now we mix it and we wait for 15 minutes for hybridization to occur. So once hybridization has been completed, about 15 minutes in, our sample is then ready for the next stage. We move on to stage 2, which is the wash procedure. This wash procedure involves our wash solution kept inside this bottle and a wash syringe provided here. It's a one-shot procedure whereby all the wash that's provided is used to clear out the contents. The wash solution provided in the bottle is taken and drawn up using the syringe. We then use the syringe inside our test here to carry out a washing procedure. This washing procedure evacuates any unhybridized material, therefore leaving the specified target bound onto the side of the tube for the later colour development stage. Now, the third and final stage, colour development. TMB peroxidase substrates provided, which will provide the colour change for the reaction, and a final syringe to draw up and then snap close the system, ready for analysis and results. Using the syringe then, draw up our colour developer, half a ml is taken, we then take our test again after having been washed. The system is enclosed via a snapping and the colour developer is added down into the tube. The test is then inserted into a colorimeter. So within a few minutes we can see that the colour developer inside is reacting and changing from a colourless solution to blue. This reaction will progress for 1 minute to 1.5 minutes, with full colour development being seen within 2 minutes. It's a kinetic colour reaction whereby the strength and rate of colour change is determined by the quantities of target that are in the sample.
Now you've seen how our test works. This has been taken to proof of principle and has been shown to detect the MRSA gene in whole MRSA down to 100 cells per mil in the University of Northampton. We've filed a patent on the detection of genes in whole bacteria without the need for extraction or purification. This is what makes the test unique and innovative and adaptable for use in point-of-care situations. You can see that our test meets many of the criteria of the Longitude Prize for a rapid, simple-to-use point-of-care test for detection of bacteria. We funded the development to date from retained income from the sales of our tests and from our shareholders' funds. However, our funds are limited in this small company and it's holding back the development of the other tests. We're seeking a Discovery Award to develop a test for the so-called nightmare bug that is resistant to virtually all antibiotics, called CPE. It is rapidly increasing in numbers worldwide. We also need some plastic moulding done for the syringes to make the test easier to use. We want to start development at Bentham Instruments of a handheld reader, which will help in the reading of the results. All of these are what we want to spend the Discovery Award on. We believe our tests will help rapidly detect antibiotic-resistant bacteria, such that it'll enable doctors to quickly decide on treatment options rather than prescribing broad-spectrum antibiotics whilst waiting for the lab to give them results, which can often take over 24 hours or more. We look forward to hearing from you. Thank you.